Good evening. Today I will talk to you about food contaminants. There is a long list of food contaminants, and covering that whole list is a Herculean task. So I'm going to cover the major contaminants for which we have limits and regulations in the Food Safety and Standards Act of India. So, what is a food contaminant? So, food contaminants are substances which come into the food unintentionally. Some of these are harmless, whereas others are hazardous because they have some toxic effects, especially when humans consume them. So contamination, if you look, of food can happen at various stages of the food processing chain. It can start from the raw material, and if it starts from the raw material, it gets carried from the farm to the fork right up to when it comes to the table. Contamination can occur during processing, where you have the cleaning chemicals, etc., which come in. It can happen during packaging. It can happen during storage. And so you see that contamination occurs at many points, can occur at many points in the food chain. So most of the time, it is the contamination in the raw materials. And the raw materials are contaminated by environmental pollutants such as the pesticides, the toxins, the mycotoxins especially, and the heavy metals, which are the major contaminants. It can also occur during processing, during filtering, and especially when cleaning of the equipment is taking place. And as I said, it can also occur during packaging and storing. That is, the plastics can leach, into the stored food or the packaged food. You could have leaching from the cans, and toxins could also be produced within the food because of the microbial contamination. I am not going to discuss anything about microbial contamination, I think, because you are going to have special webinars on food microbiology and how contamination occurs. So if you look at the food hazards, there are three types of food hazards. One is the biological, the chemical, and the physical hazards. The biological hazards come mainly from the contamination by microorganisms. The physical hazards come because of the processing, especially glass, wood, stones, pieces of plastic, etc. It is the chemical hazard, which is a very large community of hazards, and we can classify them into the naturally occurring chemical hazards or the naturally occurring toxic materials, which are allergens. We have gone through the allergens. We have the toxins, mycotoxins, which we will look at in detail. You have mushroom toxins, phytohemagglutinins or the lectins, which we have talked about the alkaloids like solanin, etc., in potato. Then we have the added chemical hazards. These are unintentional because they are added to the crop or they are given to the animal and they get carried into the food. So these include pesticides, fertilizers, antibiotics, and the PCBs. And then in the toxic metals, they generally come through the water or from the water into the food. And these involve lead, cadmium, mercury, and arsenic. And cyanide, you see, has comes mainly from the nutritional factors. The other contaminants which come in, especially because of processing, are the lubricants, the cleaning material, the sanitizers, the paints, refrigerators, etc. Refrigerants that are used, etc. So the major food components that I will discuss are the pesticide residues, the heavy metals, the mycotoxins, antibiotics and veterinary drug residues, one or two biotoxins, and bisphenol A in the packaging material. So now coming to what is a pesticide. All of you know that a pesticide is a substance or a chemical which is used to prevent crop loss by any pest. These pests can be insects, they can be rodents like mice, they could be other animals, they could be weeds, fungus, bacteria, and virus. And we know that plants and animals can be easily destroyed by any one of these pests. The pesticides include chemicals which are called as herbicides or fungicides and various other substances which are used to control pests. So insects are not the only pests. We have rodents also which are pests and which need to be controlled. So the classification of pesticides based on the genera that they inhibit or kill. We have the insecticides, which are directed towards the insects, and they have further been classified into the organochlorines or the organohalides, the organophosphorus. That means they contain the organic group with phosphorus. Example, chloropyrifos and malathion. We have the carbamates, carbaryl, carbofuran, etc. And we have the pyrethrins, which are cypermethrin and permethrin. These pyrethrins are unique in that they are actually developed based on the chemicals which are found in the chrysanthemum flower. So they are nature similar. The pyrethrins are generally in the pesticides or thing which are got from the chrysanthemum flower. Then you have the fungicides, which are targeted against fungus. So you have the dust, sulfur dust, mancozeb, copper sulfate, and these are generally used like in grapes, where the fungus thing grows. Then we have the herbicides, which are used to kill the weeds, because many a time the weeds will overtake the plant, and there will be losses to the farmer, and paraquat, alcor, etc. Then you have pesticides against rodents, rodenticides. Rodents can destroy the plants faster than the insects or the fungus. So you have barium carbonate, bromidolone, warfarin, and then you have fumigants such as aluminium phosphide, ethylene dibromide, and zinc phosphide. So classification of the insecticides can also be done based on the chemistry. So we have the organohalogen. So here you have HCH, DDT, etc. You have organophosphorus, which has the phosphorus, so monocrotophos or phosphemidone, the carbamates, the pyrethroids, the neonicotinoids, and you have miscellaneous pesticides such as the spinosins, the fiprols, pyrols, benzyl urea, and quinosalines. So these are the major 5 groups, and then you have the miscellaneous pesticides. Pesticides also can be found or available in pesticide formulations. These are mixtures of pesticides with some inert diluents and auxiliary chemicals. So the advantage of using pesticide formulations is that they are safer and easier to handle. It enables the farmer to dilute water-insoluble pesticides in water. They can get a more homogeneous and uniform application. It reduces the toxic hazards to the user, that is the farmer who is spraying. There is efficacy and selectivity, and of course there is enhanced storage stability, and it is much cheaper than the pure pesticide. Pesticides can also be uh, described as either a contact pesticide or as a systemic pesticide. A contact pesticide is a pesticide when sprayed on the plant is on the plant, and so when the insect comes, it comes into contact with the plant, it chews the material, and in addition it takes in these pesticides which are sitting on the leaves or other parts of the plant, and these include endosulfan, malathion, and fenvalerate. The second type of pesticide is a systemic pesticide. That is a pesticide which has to be taken up by the plant through the root. It has to move through the circulatory system of the plant, and it is when it reaches different parts of the plant, when the insect sits on it and eats it. So the monocrotophos or carbofuron is within the parts of the plant. Some of the chemicals have got fumigant action apart from being contact pesticides and systemic pesticides. These include DDBP, lindane, and chlorpyrifos. So how do these pesticides work against insects? This mode of action is against insects. So if you look at the major mode of action or the main mode of action, it is actually targeted towards the nervous system of the insect. So this group of organophosphates and carbamate insecticides are actually inhibitors of the enzyme cholinesterase or acetylcholinesterase, which is responsible for breaking down acetylcholine after it has conveyed the message to the synapse in the nerve, that is the nerve impulse. Acetylcholine must be broken down into acetic acid and choline so that the second nerve message can be sent to the synapse. So these organophosphates are inhibitors of cholinesterase. The organophosphate is an irreversible inhibitor of cholinesterase. We know about reversible and irreversible inhibitors. So it completely inhibits cholinesterase. So therefore, when acetylcholine is taken into the nerve synapse, it cannot be broken down. Acetylcholine will accumulate, and because of the accumulation, the insect will be paralyzed because there is no more breakdown of the neurotransmitter, and there will be the accumulation of the acetylcholine, which causes paralysis. In the case of carbamate, the inhibition is reversible, and so when all the carbamate is used up, cholinesterase will be active. So large amounts of carbamate have to be done. So the organophosphate poisoning is irreversible. That is, the insecticide actually it has got a fluoro group, and this fluoro group binds with the serine hydroxyl group at the active site of cholinesterase and converts it into a fluoride, and so therefore the serine hydroxide group is not available for action. Then you have the acetylcholine receptor stimulation. The neonicotinoid insecticides, they mimic the action of the transmitter acetylcholine. Cholinesterase is not affected by the neonicotinoids, but the nerve is continuously stimulated and there is overstimulation of the nerve, which results in the production of large amounts of acetylcholine, which leads to poisoning and death. So a closer mimic of the acetylcholine, insect's acetylcholine receptor than for human acetylcholine. That is, the carbamate, binding to the receptor is much greater in the case of insects. So therefore, carbamates are less poison, have a less ability to poison humans, unlike the organophosphates and the some of the carbamate insecticides. Then you have some of the insecticides can act as sodium channel modulators, especially the pyrethrins and the pyrethroids. I told you these are obtained from the chrysanthemum plant. These act on tiny channel, that is the sodium channels, and you know that for the cell to work properly, there must be a flow of sodium and potassium, and that is the concentration within the extracellular and intracellular has to be maintained, and it is due to a sodium channel. So this is a sodium channel blocker. So they close the channel and it eventually leads to death. So these are actually sodium channel blockers. Then we have the insecticides such as novelron, which acts as a chitin synthesis inhibitor. Most of the insects, if you see their exoskeleton, that is the outside, their skin-like structure, is made up of chitin, and chitin has to be synthesized. So these insecticides act as chitin synthesis inhibitors, and they prevent the molting of the insects, and the insects cannot reach the adult stage or reproduce and they die. The chitin synthesis inhibitors are not toxic to the humans because in the humans we do not have the metabolic pathways for the synthesis of chitin, and we do not synthesize chitin. Then you have the insect growth regulators, which actually attack the insect's endocrine system, and it produces or inhibits the juvenile hormone that is required for growth and development to the adult form. So insects which are poisoned with the insect growth regulators cannot molt or reproduce, and they eventually die. Azadirachtin, which is a natural insecticide, which is got from the neem fruit, interferes with the synthesis of the thoracicotropic hormone of the insect. Again, these insect growth regulators are not toxic to the humans because we do not make these hormones of molting. So the IGRs are considered to have little human toxicity. And finally, we have a group of insecticides which are energy production inhibitors. These inhibitors prevent the formation of energy, that is, prevent the formation of ATP. They block the synthesis of the metabolic pathway, and they prevent the insect from eating because energy is not produced. So azadirachtin, in addition to being an inhibitor of the hormone, is also an energy producing inhibitor. So how is the toxicity of a pesticide measured? It is measured by a dose known as the LD50, which stands for lethal dose 50. It is the amount of pesticide that can kill half of the animals in a laboratory test. The smaller the LD50 of the pesticide, that means the more it is toxic. And it is measured in units as milligrams per kilogram. Sometimes when it is very toxic, you will have the LD50 in micrograms per kilogram. The lethal concentration 50 is a measure of the inhalation toxicity because the pesticide can also be toxic when inhaled. So the concentration of a pesticide that causes half of the animals to die by the through the inhalation route is called as the LC50, which is measured in milligrams per liter or ppm and sometimes in milligrams per cubic meter. So how does the pesticide enter um, either the human body or into the insects? If you look at how it is toxic to humans, one is through the dermal route. That is, you have the wet, dry, and gaseous forms of the pesticide, and they can be absorbed through the skin. And the oil-based pesticides are more absorbed than the water-based. And once they get in through the skin, they enter the bloodstream and are carried throughout the body. By the inhalation route, the dust, the sprays, etc., can enter into the lungs. The particles tend to stay there and it causes irritation of the throat, nasal passages, and it then enters into the bloodstream. The oral route is through the mouth, that is by ingestion, and it can get absorbed anywhere through in the esophagus to the stomach to the large intestine, but it mainly takes place in the small intestine. So there are three major routes of entry into the human. So what are the deleterious effects or detrimental effects to humans? It can result in reproductive effects. It can result in teratogenic effects, that means in the unborn offspring. It can cause cancers of different tissue. It can cause mutations in the genetic material. It can be neurotoxic, that is poisoning the nervous system. And it could also be an immune suppressor, that means blocking the natural response to the immune system. So possible adverse effects are that it can damage the thyroid and hormonal, reproductive, and the metabolic pathways, cause cancers, liver damage, irritation to the skin and eyes, vomiting and diarrhea, and so most of these adverse effects of pesticides are seen in the farm workers who actually do not use the pesticide in the proper way. It is mainly due to the improper use that has caused these health risks. So a pesticide in any country or in the world is banned when it poses an unreasonable risk to humans or the environment, or it has a common active ingredient with any other chemical that is toxic, and it has a high exposure risk one time or multiple times. So WHO report says that there are over 1,000 pesticides which are used around the world to ensure food is not damaged or destroyed by pests, and each pesticide has different properties. Some of the older pesticides that we know, such as DDT and lindane, which remain in the soil for a very long time, that means their half-life is very long, have been banned. Several countries have signed the Stockholm Convention, and those countries which have signed the Stockholm Convention have agreed in the treaty that they will not produce or use these persistent organic pollutants, that means which remain in the soil for many, many years. So example of a persistent organic pollutant is DDT and lindane. So how are pesticides regulated in India? We have the Insecticide Act 1968 and Rules 1971. This comes under the Agricultural Ministry. All pesticides to be used in the country have to be registered, and they have a number. This act controls the manufacture, storage, distribution, export, and import, and it also controls or sets the standards for the quality of the formulation. And then we have FSSAI, or the Food Safety Standards Authority of India, in which we have the Food Safety and Standards Act 2006 and the Rules 2011. These two have a control over the residues in the food. And the FSSAI fixes the maximum residue limit, MRL, for various pesticides, which can occur in food. And then you have the Bureau of Indian Standards, which also formulates the standards and specifications for each pesticide. This is not a mandatory standard, it is a voluntary standard. And Bureau of Indian Standard develops analytical methods, and they also give the recognition for laboratory testing. So a laboratory can be BIS recognized for pesticide testing. So we have three different bodies which actually regulate the pesticides. So WHO has two objectives in relation to pesticides, and that is to ban pesticides that are most toxic to humans, as well as pesticides that remain in the environment, and to protect public health by setting the maximum residue limits for pesticides in food and water. So the risk assessment of pesticides is very similar to the risk assessment of food additives. And just like we have JECFA, here we have a joint FAO WHO meeting on pesticide residues called JMPR. The experts in these committees are responsible for actually fixing the limits or finding the ADI, or what we, instead of ADI, they will call it the total dietary intake, and indicate or assess the lab experiments to indicate what is the safe intake and exposure limit of these pesticides throughout the lifetime. So JMPR is similar to JECFA, but this is for the pesticide residues. So the limits set for this are called the MRLs, or the maximum residue limits for pesticides in food. The Codex standards are the reference for international trade in food, and these standards are set by Codex so that people all over the world have confidence in the food that they buy. WHO and FAO have developed the International Code of Conduct on Pesticide Management, and it guides the regulators, private sector, civil stakeholders on the best practices in managing pesticides. So this is actually called a code of practice. So Codex Alimentarius has this code of practice. So we have now coming up newly are the biopesticides, or the green pesticides, which are supposed to be biodegradable, safer, and natural methods are being explored, such as the use of cow's urine, plant leaves, shellac, and some yeast to thing. It is supposed that these will be are less toxic to humans and animals. And any pesticide which has the LD50 of more than 50 milligrams per kilogram, with LD50 of 50 milligrams per kilogram or less, have to have this label. It has to have this warning label, and that is the danger label, and the word poison, and it must describe the skull with this crossbones. Okay, so danger has to be there. for anything which is 50 milligrams or below. So now coming to the next biggest contaminant is the heavy metals. The term heavy metals refers to any metallic chemical element that has a relatively high density and is toxic or poisonous at low concentrations. The heavy metals are natural components of the Earth's crust. They cannot be degraded or destroyed. They enter our body through food and drinking water. They are some of them are trace elements, some are heavy elements which are essential to the human body, as we saw the other day when we did vitamins and minerals. However, the same ones can be toxic at very high concentrations and can lead to poisoning. And heavy metal poisoning could result from drinking water contamination. Actually, the lead pipes, that is, the discovery of heavy metal poisoning was because of the drinking water contamination which came through lead pipes. So the nutritionally important metals we have looked at. So they are chromium, copper, iron, magnesium, manganese, selenium, and zinc. And the toxic heavy metals are aluminium, arsenic, cadmium, cobalt, lead, mercury, nickel, and tin. So if you look at mercury, it is the most toxic of the heavy metals. There are three forms of mercury which are toxic. One is the elemental mercury, that is mercury liquid or vapor. The inorganic salts of mercury, such as mercury nitrate, and the organic metallic salts, such as methyl mercury. So the mode of entry, elemental mercury and the vapor, is through inhalation and ingestion. The transdermal, that is the methyl mercury or the organomercury, can be absorbed through the skin. And all the three can be also systemic, that is, they can be carried by injection or by thing to the brain. So the actual mechanism of action of mercury is mercury can actually chelate the sulfhydryls or the SH. So it actually prevents all the free sulfhydryls and enzymes containing the sulfhydryls at the active site from being active. It inhibits them or irreversibly inhibits them. So the on exposure to elemental mercury, you see tremors, emotional changes, there are neuromuscular changes, headaches, and there are changes in the nerve response. At very high exposure, you can have kidney damage, respiratory failure, and death. Organomercury effects, that is the methyl mercury, actually the major effect is due to exposure in the womb, that is the fetus, and it usually comes by the mother consuming fish which is contaminated with methyl mercury. And you find a large number of reports of several countries estimating the methyl mercury content of the fish along their coast, and this can adversely affect the fetus's growing brain and nervous system. So the thinking, the attention, the fine motor and visual skills are impaired, especially those children who are exposed to methyl mercury in the womb. Inorganic mercury can result in damage to the GI tract, nervous system, and kidneys. And in this, methyl mercury is the most toxic. Then you have lead. This is the elemental form of lead is not an environmental problem, that is as Pb, but when it becomes an ion and it dissolves and you have lead 2 plus, then it is highly toxic. And that is what happened in the lead pipes. And Pb2 plus is less stable than Pb4 plus. So the mode of entry is generally through contaminated water. So how does lead become toxic? It can inhibit the synthesis of hemoglobin. It has an adverse effect on the central nervous system. It interferes with the normal development of the child's brain. So you have behavioral effects and low IQ. So how does lead contamination occur? It generally occurs from when storage batteries are disposed, from cable coverings that are disposed, from plumbing, that is from the lead pipes, the disposal of ammunition and lead shots. Paints contain lead chromate and lead carbonate. So when this is disposed and when these are used in the houses as paints, they lead to inhalation or the toxicity of lead. And also, most of the soldering done in the industry uses lead, and the disposal of these causes lead contamination. Then we have cadmium, which is a soft metal. It's a bluish white metal and can easily be cut with a knife, and its compounds are highly toxic. Again, cadmium is not toxic, but the ionic form, that is cadmium 2 plus, is the toxic form. You can find this in seafood, in the organic meats which have high levels. So it is acutely toxic. At low levels, cadmium is complexed by a sulfur-rich protein and is eliminated by urination. Kidney disease may occur on constant exposure to cadmium. And excess cadmium, which is not removed by our body through this complexation process, is stored in the liver and kidneys. So where does this cadmium contamination come from? It comes because the metal smelters, they all use cadmium along with zinc, lead, and copper. It comes from burning of coal, and it comes from incineration of waste containing cadmium, such as plastic, batteries, etc. Arsenic exists in different states, and it is the arsenic in the trivalent form which is most common and most toxic. It occurs as arsenic trioxide, organic and inorganic. It mainly comes through contaminated water, and WHO had published a report on the high levels of arsenic contamination in the water of Bengal. And people drinking there, most of the deaths were due to the highly contaminated water of arsenic. Even now, the whole of the Ganges belt, you will find the arsenic levels of the water are high, resulting in very high levels of arsenic, especially in basmati rice. So the ingestion is mainly through contaminated water. The arsenic oxide, the trioxide, can be absorbed through lungs and anything. It is a known carcinogen and causes gastrointestinal damage. It coagulates proteins and complexes with coenzymes. So therefore, it doesn't allow the metabolic pathways to proceed. It inhibits the production of ATP, and arsenic 3, that is As2O3, arsenic in the 3 form, 3 plus form, is more toxic than arsenic in the plus 5 form, presumably due to stronger binding with the sulfur containing proteins. So this is how arsenic causes the toxicity or mode of action. So here you have arsenic compound, and you have a protein or an enzyme with a sulfhydryl group. It is the heavy metal here which will chelate these two sulfhydryl groups So you see, it chelates the two sulfhydryl groups. You have an inactive protein, and the protein cannot work. So therefore, many enzymes which have got sulfhydryl groups at their active site, many proteins which have got sulfhydryl groups are inactivated by arsenic. It is structurally analogous to phosphate and is attached to proteins by binding to their SH groups. So these SH groups can either be cysteine, or it could be dimercaptosuccinic acid, or it could be glutathione, or to proteins such as hemoglobin, metallothionein, and thioredoxin, all of which contain sulfhydryl groups. So arsenic lethal effect when consumed in an acute dose, it causes gastrointestinal disturbances. So as we said, arsenic in the 3 plus form is the most toxic. So you see the LD50 for that is 14, whereas for arsenic acid it is 20. And as you see, the higher it becomes less toxic. And if you look at the toxicity of some of these metals that we have looked, it is the speciation of the toxic metals which cause their toxicity. So we have seen in mercury, rather than mercury, it is methyl mercury which is more toxic than the elemental mercury or the inorganic mercury. Lead, the soluble ionic form Pb2 plus, is more toxic than earthing lead, and arsenic in arsenic oxide is more toxic than arsenic. So this ionization is known as speciation of toxicity in metals. Then you have chromium also, which is toxic. Chromium can exist either as trivalent chromium or as hexavalent chromium. So if you look at the toxicity, chromium is a toxin. So this is a suspected carcinogen in the 6th form, whereas the chromium 3, which is less toxic, actually acts as a trace element. So it is the chromium 6 which is toxic, and so that is why many of you will be using chromic acid for washing your glassware. Actually, using of chromic acid for washing glassware is banned. So please do not use chromic acid to wash your pipettes and other glassware. So sources of contamination come from the leather tanning industry, from metal plating, from the leaching in hazardous waste sites, from wood, and unintentional release during the mining of and smelting of gold, copper, and other metals. So artificial methods to remove heavy metal toxicity or heavy metal poisoning is done by chelation. So the chelators that are generally used to remove heavy metals are cysteine. If you recollect the structure, it has a CH2SH as the side chain. Then you have EDTA, ethyl diamine tetraacetic acid. So you see here you have a metal. So the metal is chelated by the tetraacetic acid of EDTA. You have citric acid. So citric acid also can chelate metals, and plant phenolic compounds such as the tannins, etc., also are used to remove the heavy metals in different foods. So if you look at the safety assessment of the heavy metals, the heavy metals safety has been established by JECFA Codex, and they use a term TWI, which is defined as the amount that an individual can ingest over weekly over a lifetime. ADI is acceptable daily intake. PTWI is the accepted weekly intake over a lifetime without appreciable risk, and it is expressed in milligrams per kilogram body weight per week. So if you look at the toxicity of the metals, if you look at methyl mercury, it is 1.6 micrograms per kilogram, very toxic. Then you have cadmium, which is 7, followed by arsenic, lead, nickel, and these are at much higher concentrations. So that is why, somebody was asking me, how do they set the limits? The limits are set based on the um toxicity of the elements and how much of the food that we consume. So in a week, you cannot consume more than 1.6 micrograms of methyl mercury. If you do that, it will become toxic. Then you have some biotoxins. These biotoxins are poisonous substances that are actually produced by metabolic activities of living organisms, could be either plant, animal, bacteria, or fungus. They cause food intoxications, and the dose depends on the toxicity depends on the dose. So the toxin classification, if you look at the bacteria, which produces, one of the major toxins produced is botulinum toxin or staphylococcus toxin. These will be covered in more detail in the microbiology section. Fungi produce or toxins which are called mycotoxins, and they include aflatoxin, ochratoxin, patulin, zearalenone, deoxynonobinol, and there. Algae also produce toxins like okadaic acid. These toxins are generally consumed by the fish, and it is through the fish that they come into the food chain. So just to tell you about botulinum toxin, it is produced by Clostridium botulinum, a gram-positive organism. It is an anaerobic spore-forming rod. The toxicity you see is 1 nanogram per kilogram. 500 grams is enough to kill the whole human race. So therefore, actually, the botulinum toxin is used in biological warfare. And botulinum again interferes with the acetylcholine esterase mechanism at neuro junctions, and this results in muscle paralysis. It causes nausea, headache. But you see, it causes muscle paralysis. So therefore, it is actually being used in beauty parlors, especially for face uplifts and to remove wrinkles on the face. Now let's come to the mycotoxins, which are very important. This word mycotoxin stems from the Greek word mykes, which means mold, and toxin means poison. The mycotoxins are secondary metabolites which are produced by these fungus in the plants they use as food, and they contaminate the agricultural produce. The mycotoxins can either be pre-harvest or they can be post-harvest. That means they grow on the plant, and the toxin is present right from the beginning, or the spores, etc., are there, and on storage, on post-harvest, the conditions are conducive for their growth. So in the mycotoxins which are associated with food and feed, the most important are the aflatoxins, that is B1, B2, G1, G2, and M1, ochratoxin, zearalenone, the fumonisones, DON, that is a trichothiazine, patulin, sterimatocystin, which is a precursor of aflatoxin, and you have cyclopioxonic acid, which is got especially caused by the infection of alternaria. Okay, and these are miscellaneous ones, and I will just tell you about these and what our regulations think. So the mycotoxins are chemically diverse and they're toxicologically diverse also. Among the aflatoxins, aflatoxin B1 is the most toxic, and it has been recognized or grouped under a group 1 carcinogen. The ochratoxin is a potential carcinogen, which is under group 2B. Fumonisin is toxic to the liver and the kidney. Zearalenone is a skin and membrane irritant. Patulin, which is found in apple juice mainly, is genotoxic, and citrulline is nephrotoxic. So, which are the organisms which produce these? Aspergillus parasiticus produces B1, B2, G1, and G2. B2 is the most toxic. Aspergillus flavus produces B1 and B2. Fusarium sporotrichoides produces T2. The Fusarium graminearum produces nonanivol, which is DON. Fusarium moniliform produces zearalenone. Aspergillus ochreus produces ochratoxin, and Penicillium verrucosum produces fumonisin. So the crops which are generally contaminated with these mycotoxins are peanut, maize, almond, fig, most of the nuts, the seeds, and you will see that even rice and chili powder. Aflatoxin comes into milk because the cow, the feed contains a large amount of aflatoxin B1, B2, G1, and G2, and that gets converted into aflatoxin M1. Ochratoxin is found in coffee mainly, grape, pepper, etc. The fumonisins are found in cereals, so also the trichothecenes and the zearalenone, and ergot alkaloids are got when the ergot fungus actually infects rye, barley, wheat, and oats. So we have what is known as primary and secondary mycotoxicosis. The primary mycotoxicosis is the infection of the produce over there. So any one of these produce are there. So direct consumption of peanuts or the cereal grains or the oil seeds which are infected with the mycotoxin, when injected directly, we can develop primary mycotoxicosis. Secondary mycotoxicosis occurs when the feed of the animals contains the mycotoxins and we consume either the animal or the products of the animal, and that causes secondary mycotoxicosis. So aflatoxin, there are 4 types of aflatoxin. It is one of the most potent carcinogens, that is of the liver. It asserts acute and thing. You have B1, B2, G1, and G2. Their names B and G are got because they exhibit blue and green fluorescence. So G exhibits green fluorescence and the B exhibits blue fluorescence. So they're crystalline, they are freely soluble in polar solvents such as chloroform, methanol, etc. They exhibit, as I said, blue and green fluorescence, and aflatoxin M1 is a metabolite of the B1, and M2 is a metabolite of B2. So acute toxicity, you see, the LD50 is very low, showing that it is toxic. So you see it is toxic to almost all the mammals. Okay, several methods are there to assay it, but nowadays we use the HPLC and we use the LCMS-MS to detect the alpha toxins, and you can even go down to as far as zero 0.1 nanogram. And now coming to ochratoxin, this damages and causes cancer of the kidneys. It's a group 2B carcinogen. It is associated with nephropathy, and the total dietary intake is set at 5 nanograms per kilogram body weight. So ochratoxin is as toxic as a little less toxic than aflatoxin. Patulin, this is a genotoxic toxin, and FSSAI has put a limit only in apple juice where it cannot exceed 50 micrograms per kilogram. It exhibits strong antibiotic activity against bacteria. The fusarial toxins such as the fumonisins, the trichothecins, and also are toxic. So you see fumonicin, the TDI is 2 micrograms. The trichothecins, it is 1 microgram. And for zearalenone, it is 0.2. So therefore, the presence of these contaminants is regulated, and several of the commodities have been, the limits are there. Sterimatocystin is a very recent entry into the toxic group of mycotoxins. It is known to be a precursor of aflatoxin B1. So you have sterimatocystin, which can get converted into aflatoxin B1. This also is carcinogenic, and it can be found in spices, grains, bread, beer, and cheeses. So this is the FSSR limits. So you see aflatoxin is regulated in cereal and cereal products, should not exceed 15 micrograms, pulses, all nuts, dried figs in ready to eat, it is 10, dried figs 10. If you look in spice, it is a little more. And see, the genesis behind this is, if you look at nuts, we eat plenty of nuts. So therefore, the average daily intake and the exposure to this will be much more than spices. Spices are used in very minute quantities in different dishes. We don't eat grams and grams of spices. So therefore, you see the limit in spices is much higher than the limit in nuts or pulses. So the same is the case with the additive. I'm explaining this to you because I had somebody ask me this question. So why is it different in different commodities? So it is based on how much of these commodities we eat. So you see, we eat a lot of dried figs. We don't stop with one. We can have more and things, but we cannot eat more than 1 gram of spice. So that is the reason of this. Aflatoxin M1 in milk is 0.5 microgram. Ochratoxin is for these three. Patulin is mainly in apple juice, and DON is given in wheat. So now coming to the veterinary drug residues and the antibiotics, they have got both benefits and risks. The benefit is they are used for animal health and welfare. In human nutrition, when we are sick, we take antibiotics. However, the risks are again for the animals, for humans, if we develop antibiotic resistance by consuming too much of antibiotics. So, if you look at the veterinary drugs and the antibiotic residues, the veterinary drugs are generally used in the farm animals in the form of injectable doses or as dermal doses, or they are fed directly to them as oral tablets or a soluble powder or granule feed. So from the animal, it comes into the animal product, which is either the meat or the milk. And then it is the human who eats the meat and the milk. So indirectly, the residues enter the human body. So substances that are used in animal feed and have got anabolic effects are the stilbenes, the antithyroid agents, the steroids such as androgens, gestrogens, and the estrogens, ricinoleic acid, lactones, the beta agonists, and other compounds. So all these are used for some purpose or the other. The antithyroid agents as use the thiocertine, it is to prevent or inhibit the formation of the thyroid hormone thyroxine, because thyroxine is involved in the basic metabolic rate. It increases the basic metabolic rate of the thing. So in order for the flesh to grow, that's for a chicken or for an animal, if an antithyroid is given, there will not be too much of metabolic activity, and so the muscle will increase. So all these have got different properties, and these are used. So coming to the antibiotics, we know that it was Alexander Fleming who discovered the first antibiotic, penicillin. So the antibiotic can be defined as a chemical produced by a microorganism that kills or inhibits the growth of another microorganism. An antimicrobial agent, on the other hand, is a chemical that kills or inhibits the growth of microorganisms. It is not produced by the microorganism. That is the difference between an antibiotic and an antimicrobial agent. So the microbial sources of antibiotics are produced by either the gram-positive or thing. So you see, you have bacitracin, polymyxin, you have the actinomycetes can produce erythromycin, streptomycin, gentamicin, chloramphenicol. The fungus which produce the antibiotics are Penicillium notatum, the first one Penicillium, and you have cephalotone. So this is to tell you that the antibiotics are produced by the microbial sources. And each of these antibiotics has a different spectrum of activity. So the antibiotics can either inhibit the gram-positive bacteria, or they can inhibit the gram-negative bacteria. There is no single antibiotic which can inhibit all the gram-positive and all the gram-negative. If anyone discovers such an antibiotic, I'm sure it deserves the Nobel Prize. So an antibiotic is again, the spectrum varies. So you see, for example, penicillin works on gram-positive bacteria. Okay, tetracycle can work on some gram-positive and some gram-negative. So the bacteria, when they are growing, they have their own enzymes which are required for cell wall formation, protein synthesis, DNA replication, RNA synthesis, and synthesis of essential metabolites. So most of the antibiotics will inhibit one of these processes. So they will inhibit either cell formation, cell wall formation, or protein synthesis, or DNA replication, or RNA synthesis, and thus inhibit the um, growth of the microorganism or the microbe. So modes of antibacterial action are: it can prevent transcription, that is the transcription of DNA to mRNA. They can also inhibit the cell wall synthesis. Examples are penicillin, cephalosporin, bacitracin, and vancomycin. There are antibiotics can inhibit protein synthesis. That means the translation of mRNA into the protein. These include chloramphenicol, erythromycin, tetracycle, and streptomycin. They can also inhibit replication and transcription. These include quinolones and rifampin. especially rifampin, which is used in tuberculosis to prevent tuberculosis. There are some which will not inhibit cell wall synthesis, but they will injure the plasma membrane. That includes the polymyxin. And then you have some antibiotics which inhibit the synthesis of essential metabolites such as sulfonylamides and the trimethoprim. The sulfonylamides are known as the sulfa drugs. So the inhibitors of cell wall synthesis known over several things are penicillin, which different forms of penicillin are known. So the active part of penicillin is this beta lactam ring. So common ampicillin, many of you might have taken ampicillin. It is also from the penicillin group, and it has this beta lactam ring. It inhibits cell wall synthesis. Then you have others such as cephalosporin, bacitracin, vancomycin, and which also have the nucleus which is similar to the beta lactam ring. You have inhibitors of protein synthesis, that is chloramphenicol, aminoglycosides, tetracyclines, and the macrolides. Inhibitors of nucleic acid synthesis, rifamycin, which again, as I said, is a major anti-tuberculosis drug. You have quinolones, examples, ciprofloxacin, which inhibits DNA gyrase. And you have the sulfonamides, which are competitive inhibitors of several enzymes. And you know what are competitive inhibitors and non-competitive inhibitors. So the following antibiotics and veterinary drugs are not permitted to be used at any stage of processing of meat or meat products, poultry and eggs, seafoods, including shrimps, prawns, or any variety of fish and fish products. The maximum residue limit is 0.001, and for chloramphenicol is 0.003. These are actually limits of the instrument. So you see here, nitrofurans are not permitted, chloramphenicol is not permitted, colchicine is not permitted, and then you have clenbuterol and diethyl stilbol, which are anabolic compounds, and you also have the crystal violet and malachite green, which are antifungal compounds, which are not permitted. So all these antibiotics and veterinary drugs are not permitted in the meat. And then we have a list of 103 antibiotics, veterinary drugs, and residues which are regulated by FSSAI. FSSAI has given the limits, that the limits cannot exceed. It varies in meat, poultry, milk, and other products. I have only listed a few because I cannot list all the 103, but you can see the recent regulation of January 2019. Then we have another contaminant which everyone talks about, which is bisphenol A. This is a chemical which is found in hard plastics that we use every day, including water bottles, baby feeding bottles, dental fillings, eyeglasses. It is known to be an endocrine disruptor, and it is said that children are the most sensitive, and research suggests that it can impact human health in various ways also. Therefore, the only remedy is to use bisphenol A-free products. And now many manufacturers are creating the BPA-free products. It cut back on the using of cans and plastic, avoid heat. The National Institute of Environmental Health advises against microwaving polycarbonate plastics or putting them in the dishwasher, because the plastic may break down and allow BPA to leach. And it also suggests to use alternates such as glass, porcelain, or stainless steel containers for hot foods and liquid foods. So The next seminar tomorrow, or the webinar tomorrow, will be on food adulteration.